A very warm welcome, students. Today we are going to study about P53. P53 is very important gene in the world of biology. It is included in cell biology, cancer biology, and as well as molecular biology because it is guardian of genome. What do you understand by the word guardian? Guardian is someone who guards us, right? Like our parents, they guards us. So something which is guarding the entire genome is known as P53. When we study about cancer biology, there are two types of gene we really focus upon. First, tumor suppressor gene, and second is proto-oncogene. When we talk about tumor suppressor gene, as the name suggests, what is tumor? It is cancer, uncontrolled group of cells, undifferentiated cells group, right? Which is known as cancer. When we talk about suppressor, it is repressing something. So, what does the entire name suggest? The gene which is repressing the formation of tumor. Basically, the gene which is saving from cancer is known as tumor suppressor gene. There are four types of tumor suppressor genes. Basically, P53, retinoblastoma, P10, P63. Among all four, P53 is most important because it regulates the function of other genes also, and it is guardian of genome. Why I am repeating again and again this term guardian? We will see when we get to know the functions of P53. Okay, so reading basic points of P53, it is found on chromosome number 17. Gene which produces this protein is tumor protein 53, TP53. Why 53? Because its molecular weight is 53 kd and it is present in P53 nucleus, right? So it is present in nucleus. So it became nuclear protein as well as transcription factor. This is important point again. Fine. Now there are three cases in which we can understand the function of P53. First case is when cell is in undividing state. What do you understand by undividing? That cell is in quiescent phase. We know in quiescent phase cells do not divide. They are just resting for a while. So in undividing state, P53 is bounded by MDM2. Now, first of all, you need to know that P53 is always present in cell, but in inactive form. Why in inactive form? Because this MDM2 is negatively regulating P53. Very important point. MDM2 is inhibitor of P53. It is bounded with P53 until it is required in cell. Okay? And when it is required in cell, what do you understand by guardian again? Our guardian protect us when we are in danger. So well, and when any cell is in danger, any damage is occurring, then P53 is required. Until then, it will be binded with MDM2, which will negatively regulate P53. So now cell is not dividing. When cell is not dividing, then there is no chance of damage, right? When there is no division, then obviously there will be no damage. So we do not require P53 here. So what happens? MDM2 and P53 are binded with ubiquitin, and this ubiquitin degrades P53. So in undividing cells, what happens? P53 is degraded because here cells are not dividing, so no need of P53, right? When we go on case number 2 is of normal dividing cells. When we talk about normal dividing cells, Growth factors is increased. It follows RAS map kinase pathway. We know this is signaling pathway, right? Signaling pathway for cell division. It increases CDKs. We know cyclins and CDK increases and promote the cell division. Now, RAS map kinase pathway produces cells from Q0 phase to G1 phase. We know when cell enter in G1 phase, it is in dividing phase. Now cell will divide. But here I have mentioned normal cell division. That means no damage is occurring. When damage is not occurring, P53 do not functions. Okay? Because it is guardian again. Guardian comes to protect when we are in danger. Cell here is not in danger. But still, what will we see here? We will see that MDM2 is binded with P53, but ubiquitin is degraded. So that if in case there any damage occurs, P53 can be ready. P53 is on standby here. In first case, we studied that P53 is degraded because there is no cell division, so there is no use of P53 at all. But here, because cells are dividing, so there may be possibility that damage can occur. So we have to keep P53 on standby. So what happens here? USP10, which is doing deubiquitination, degrades ubiquitin and allows MDM2 and P53 to be on standby, right? Here, right now, we do not need P53 because there is no damage, but we are keeping P53 on standby bounded with MDM2. So, what we learned, P53 is degraded when cells are in undividing state. But when cells are dividing normally, P53 is not degraded. It is kept on standby bounded with MDM2. So, in case damage occurs, it can go and help to repair that damage. Now, here comes the case number 3, which is most important case, which will tell us entire functioning of P53. Now, case 3 says cells are dividing and damage is occurring. Cells are abnormally dividing. So when damage occur in DNA, like double stranded break, single stranded break, kinases are activated. Apical kinase, downstream kinase. These kinases signal to activate P53. So what happens when double stranded break occurs? ATM is activated, which activate check 2 by phosphorylation, and when single stranded break occurs, ATR is activated, which phosphorylate check 1, and these check 2 and check 1 phosphorylate MDM2. Now MDM2 is phosphorylated, it releases P53. Now P53 is neither bound with ubiquitin or MDM2. So what happens? P53 is now released, and released P53 is known as activated P53. Now P53 knows that some sort of damage is occurred, whether it is single break or double break. So what will the first step? It will firstly stop the damage, then try to repair it, right? So firstly, it will stop the damage. How? It will activate P21, which will inhibit CDK2. And if you know, CDK2 is responsible for G1 phase. Now, if CDK2 is inhibited, then G1 arrest occurs. Then it will activate GAD D45, right? GAD D45 inhibits CDK1, which is responsible for G2 phase. So it will arrest cells in G2 phase. And also it inhibits CDK2 and again G1 arrest. Basically, it is arresting cell in the phase cells are right now. Okay, now it is stopping DNA damage. Now damage is stopped, then DNA repair enzymes comes in action. DNA repair enzymes will then repair the damage and cells will further move forward. But in case when very severe, break is very severe, then P53 is increased in high amount. And this highly increased P53, activated P53, signals pro-apoptotic factors to do their function. Pro-apoptotic factors like Noxa, Puma, Bax kills cells. Cell move into apoptosis and those damaged cells are removed. Okay, so I hope you understand the entire functioning of P53 in all the cases. Thank you. Have a good day.